Vanakkam. Welcome to this course on classics in neuroscience. We are discussing classic Nobel Prize winning works by various scientists. In this series of videos, we are discussing the classic Nobel Prize winning works of Santiago Ramon y Cajal. In this video, we will look at Cajal's critical modification to the Golgi method that made his approach or his modification a lot more successful, that made the Golgi method a lot more reliable and not capricious or whimsical, and how his brilliance in deciding to study embryos to enable better visualization actually paid off in a very big way, and how he studied the various subsystems within the nervous system, like cerebellum, retina, hippocampus, pyramidal system, cortex, various systems or subsystems within the nervous system buttressed or supported his ideas in a huge way. Recall that in the previous video we said that Cajal is this rigorous experimentalist who knew that he needs to exercise a lot of patience to refine the Golgi method. There are only two reagents, but you need to control for concentration, you need to control for amount or volume, and you need to control for duration of the bath. These are the various factors that you could control. Cajal's careful approach to experimentation and his enormous patience, by which he changed exactly one variable at a time, and he observed the changes and he documented it very, very carefully, and he drew the outcomes because he was also a brilliant artist. He drew what he saw, and his drawings were so good that they were almost like photographs. Remember, this was the age when photography was just picking up, and taking a photograph through the microscope at that time was not practical or possible. So, he was looking at the microscope and drawing the pictures. Cajal decided that he can increase the reaction intensity by staining the tissue with silver nitrate 2 times or maybe even 3 times and for a longer duration to improve the quality of the stains. This method went on to be called as the double impregnation method. This is the double impregnation method, meaning that you stain the nervous tissue for 2 times or sometimes even 3 times and for longer duration to improve the quality of the stains. After several months of trial and error, my suspicion is that it was about 6 to 8 months of trial and error, the most elusive cells among the nervous cells started showing up, started manifesting, and they were brilliantly stained. Nobody else could produce such stains. That was because nobody else had the patience that Cajal had. Nobody knew how to stain this as much as Cajal did. That is the reason that the most elusive cells among the nervous cells started showing up only to Cajal and in his microscope. Every time he made an observation, Cajal verified his observations with Golgi's masterpiece, which is called as On the Fine Anatomy of the Nervous System, which is written in Italian. But Cajal said that someone who is well read in Spanish could practically understand Italian. So, he read the Italian book to verify whether what he found was either concurring or opposing Golgi's point of view. Golgi claimed that there is certainly to be found a very widespread network of filaments anastomosing with one with the other, anastomosing with each other. Well, are we sure that the neurons are anastomosing with one another? But Golgi made this bold claim, and remember, that was the prevalent theory of the day for 50 long years or perhaps longer. People believed that neurons were part of a fine net, a reticulum, a very large net similar and comparable to the vascular system, is what people believed, and neuronal cells and their filaments anastomose with each other was the prevalent dominating dogma of the age. But he said further, some of the finest filaments are yet to be seen. That is, these filaments, for example, those that are disappearing here, here are not seen because maybe because we really don't have the magnification. Once we have the magnification, we can see those anastomoses. And fibers that join the reticulum, once they join the reticulum, they lose any sense of individuality. These are the claims that Golgi made. Suffice to say that Cajal was thoroughly disappointed with these kind of theories. Cajal was not at all happy with this, because for one, he was not convinced that if you have non-individual neurons, then the entire system is one, but how is it that we are learning? So, recall that he was this keen observer, but he was also quite philosophical and sort of polemical in his thinking. He was this rebel in his thinking. So, he asked the question, if indeed all the neurons have no individuality, that means that the entire nervous system has one identity. In that case, how is it that we learn things, we adapt? It is not possible for us to learn if the entire nervous system has a single identity or a single individuality. That is one philosophical argument. Besides, his own observation showed that the fibers were not anastomosing with each other, but rather were independent. So, Cajal remained unconvinced about Golgi's reticular theory. But one problem was that when he studied adult nervous system and adult neurons, what happens is that these neurons are usually myelinated, they are very thick, and myelin means lipid or fatty cells that cover the axons. There is a purpose for this. There are several purposes that are now understood, but these cells obfuscate or confuse or otherwise make it very difficult to stain and understand neuronal structure. So, Cajal came up with this brilliant insight where he said, how about we study embryos? If you study embryos, that is, if we view the natural sequence in reverse, we should hardly be surprised to find that many structural complexities of the nervous system gradually disappears. That is, the complexities of the nervous system start manifesting as the embryo goes on to grow, born, and then grows on to become an adult. That is about when the complexity is at its highest. But in an embryo, much of the complexities do not exist. This is both the answer and the question, because if the complexity disappears when you go back in time, or the complexity appears as you go forward in time, that means that the structure of the system changes as a function of time, right? That is what it means. that is essentially already a disproval of a disproving point against the reticular theory. But anyhow, this is one argument. The other is that if we can study simpler systems, those that are not myelinated, maybe you will find better stains and you will be able to observe the system and the finest details better, because embryo cell bodies are simpler cell bodies, they have shorter fibers and their relationship with other cells are much easier to observe. There is no myelin. Younger axons usually do not have much myelin and they more fully absorb the stain, because myelin is not preventing them from observing the stain. Since full grown forest turns out to be impenetrable and indefinable, why not revert to the study of young wood at nursery stage? Why not study small plants, seedlings that are just growing? Why not study them? These are words within quotes, are all the actual words of Cajal. So, he started studying the avian cerebellum and he could make slices that are as small as 15 microns. Remember that Cajal actually went on to become a doctor and a professor later, but earlier he was training to be a barber physician. His father was first a barber physician, so he knew how to use the razor. So, he could make cuts as small as 15 microns, such finesse with his hands. So, when he studied the avian cerebellum and the Purkinje cell axons, what he observed was that these axons, although intertwined in a very complex fashion, he found that they actually ended freely. He repeated this test many times because he wanted to be 100% sure, because he was challenging the dominant theory of the day, the dominant dogma of the day. He needed to be 100% sure. So, he studied this with many samples, in different birds, in different settings, in different ages, and all that. He practically found the same result. Remember, Cajal said that one must achieve total absorption. You can take it as a total absorption of the stain or total absorption in your studies. That is another way in which you can take it, but this is what he said: one must achieve total absorption. He actually had total absorption in his studies, but Cajal also had to show this in other neurons, not just in cerebellum. So, he turned to retinal cells. Remember, cells of the cerebellum and retina are some of the most well organized cells in the entire central nervous system. So, he turned his attention from cerebellum to retinal cells, and what he found he describes as follows: we have never been able to see an anastomosis between ramifications of distinct axons, nor the filaments emanating from the axons, that is the collaterals of the axons. The fibers are interlaced in a very complicated manner, engendering an intricate and dense plexus, but never a network. Every time the impregnation has been fine and complete, we have only seen free nerve endings. That means that there is no anastomosis. There is never been able to see an anastomosis, and what we have seen are free nerve endings. That means neurons are separate, neurons are structurally and perhaps even functionally separate and independent. And he published 43 papers in a span of 4 years, where he reported results from his studies of birds cerebellum. Avian cerebellum means bird, aves means birds. aves. Aves means birds. Avian cerebellum means cerebellum of birds. Avian cerebellum, avian retina, frog muscle spindles, muscle fibers of the heart, optic lobe, optic nerve of birds, spinal medulla of birds and mammals. Everywhere he saw only independent cells. Not in one place he saw networks. After all these studies, by about 1889 or 1890, he was absolutely convinced that neurons were indeed separate cells. Neurons also obey the cell theory, and neurons were independent structurally, perhaps even functionally. So, he said that by this time I nourished the certainty that I was not mistaken. That is, by this time he was convinced that he was indeed right in terms of believing that the neurons are indeed independent and separate both structurally and functionally. So, with this we have almost reached the end of this lecture. In this video we saw the critical modifications that Santiago Ramon y Cajal made to the Golgi method. What are these? Double impregnation, or maybe even triple impregnation, longer duration of bath in silver nitrate. Also, he took simpler systems to study, such as the embryos. As a child, he loved collecting bird eggs. Among the many things that he did as a child, remember, he wanted to be an artist and was not very keen on the regular education. So, he used to collect bird eggs. So, after several years he again started collecting bird eggs, except that this time he started doing it for his own research. By employing simpler systems and studying embryos, he theorized that he could achieve better visualization, and he did achieve better visualization because the myelination was not there. And he studied various systems: avian cerebellum, avian retina, frog muscle spindles, frogs, various other systems, spinal cord circuitry, very different systems, subsystems within the central nervous system. Based on all these things, he was almost certain that he was not wrong, or he was almost certain that neurons indeed are independent and separate. With this, we come to the end of this lecture. Thank you very much for your attention.